Da skal jeg få lov til å ønske velkommen til dette webinaret om ME og soppinfeksjoner. Then I'll switch to English, and I'd like to say welcome to Katharine Seton and Simon Carding, who come from the Quadram Institute in Norwich in the UK. And I'll give the word to them and let them introduce themselves and give the presentation they have prepared for us. So should we start with you, Katharine?
Hi everyone, I'm Katharine Seton. I'm a postdoctoral research scientist at the Quadram Institute, working in Simon Carding's group in the ME-CFS team.
Good afternoon, I'm Professor Simon Carding. I work at the Quadram Institute, and with Katharine, we are aiming to develop a Centre of Excellence for ME research. So I am going to give a little bit of background to the work we're doing at the Quadram and talk a little bit about our work looking for the infectious causes of ME-CFS. And then Katharine will talk in particular about fungal infections and a request for help from all of you to complete a questionnaire as a survey of fungal infections in ME-CFS patients. So, next slide. So, as I said, we are at the Quadram Institute trying to develop a Centre of Excellence, and this is done in collaboration with the Invest in ME Research UK charity, which many of you may be familiar with. The goal of the centre is to establish a pathway by which we can uncover what causes the disease in order to develop the best approach to intervene to treat and hopefully cure the disease. So mechanistic studies leading to clinical trials. So our fundamental research is focused on viruses and viromes in ME-CFS. We also have projects related to looking for evidence of autoimmunity and immunodeficiency in ME-CFS sufferers, and in particular immune senescence, which is the aging of the immune system and whether or not that's accelerated in sufferers from ME-CFS. We are also undertaking translational research. Katharine has just completed a photobiomodulation assessment in patients, and we are planning on microbiota targeted therapies, and in particular microbiota replacement therapy or fecal microbiota transplantation as another means of intervention in sufferers of ME-CFS. We don't work in isolation. We work with colleagues across Europe through the European ME Research Group, or eMERGE, which I currently chair, and has members from many countries, including Norway. And we work closely with the European ME Patients Association, EMEA, and we've also established a young eMERGE, which Katharine currently is the chairperson of, with the aim there of encouraging and promoting early career researchers to take up a career in ME-CFS research. And Katharine and I are both involved in working with colleagues in the United States, particularly the National Institute of Health. Currently, we're working on improving the design of clinical trials and also identifying appropriate outcome measures for use in clinical trials. And in terms of advocacy, I've currently been involved in trying to help the UK government in establishing a research roadmap and a care roadmap for sufferers of ME-CFS in the UK. Similar organisations we've contributed to, Katharine and I, in the United States. And we're also seeking to obtain funding from UK as well as European-based organisations, as well to support research across Europe, Scandinavia, not just in the UK. This is currently the research team at Quadram Institute. It's very small, Katharine and myself, and then Krishani, who's supported by a Luna Nova Fellowship that's provided support through Invest in ME Research. And Rick, who was a former PhD student, is now a postdoc with Quadram, but also has an appointment at Amsterdam University. And he's just received a large grant from the Dutch government to undertake a study looking at the viromes in ME-CFS. So this is the central question we are trying to address in all our research at the Quadram Institute. Are infections a cause of ME-CFS? And if so, what are these infectious agents and where do they come from? Are they acquired from the environment, or they're actually residing in the body and then get reactivated to cause disease? And a lot of our studies do include the most severely affected individuals. We do try and make this an inclusive approach to involve patients and individuals with all the severities of ME-CFS. So I've used some slides here provided by Trude to sort of emphasize a couple of points. This is a European-wide patient survey which identified infections as the main trigger, a pre-infection, the main trigger of disease. See, over 50% of patients in Scandinavia, in the UK, and also in the US identified infections as the primary cause of their disease. And this is the same across all severities of disease, where infections are linked to causing severe, all the way from severe to better than mild, although in recovered individuals, this is slightly less than 50%. So clearly patients relate to an infection as being a causal factor in their condition. So what are the origins of infection? The first recorded accounts of something that could resemble ME-CFS dates back to the late 19th century, the Russian flu pandemic, where it was termed post-influenza nervosa. And this outbreak led to more than 1 million deaths. And then early 20th century, we had the Spanish flu pandemic, where we had the post-influenza encephalitis lethargica. And again, this was associated with very large numbers of deaths. Then as we move into the 1930s and 50s, we have a disease which is associated with poliomyelitis, but slightly different from it. And this has been called epidemic neuromyasthenia in the US and parts of Europe and Australasia. And then the Acuri disease occurred in Iceland, leading to it being named the Iceland disease. Then in the 1950s and 60s, there was a major outbreak across many countries, and one in particular in London at the Royal Free Hospital, which is a nurse's home that affected a very large number of people, originally designated as mass hysteria, but this was the first description of something resembling ME as benign ME. And then in the 1970s and 80s, there were outbreaks, famous outbreaks now in the history of ME-CFS in the UK, New Zealand, Canada and Ireland, and in particular in Lake Tahoe in Nevada. And this led the CDC in the USA to first use the term chronic fatigue syndrome. And then in the 1990s, it was unfortunately given the acronym yuppie flu. And then in the 2000s, we've had more outbreaks, but increasingly now it's being referred to as ME-CFS. So a lot of these other derogatory terminology have thankfully fallen away. And more recently, there have been an increase in the number of cases of ME-CFS, or patients reporting with symptoms similar to ME-CFS in those infected with SARS-CoV-2, with as many as perhaps 30% of survivors eventually developing chronic fatigue syndrome or ME-CFS. And in 2019, there was a 15-fold increase in the diagnosis of ME-CFS that was attributable to a post-COVID-19 infectious syndrome. So this slide and graph was provided to me by Trude, and I think it really shows across 4 different surveys in Norway, the peaks in incidence are coincidental with outbreaks of influenza virus pandemic, so H3N2 around 2000, and then 2 outbreaks of swine flu occurring in around 2009 and 2012, and then of course the COVID-19 outbreak where we're seeing a very large spike in new cases of ME-CFS. And we're unlikely to see the full impact of the COVID-19 pandemic until maybe a year or 2 years from now. So in terms of our outputs from the group, although we're a small group, we do publish quite a lot. We've had publications describing technologies, techniques that allow us to investigate viruses, for example. And these 2 publications relate to the analysis of human stool samples, patient stool samples looking for viruses. And then we've written some sort of articles to try and highlight the importance of viruses and the virome. Katharine herself has contributed to the outputs in looking at production of antibodies against particular gut microbes that seem to be more prevalent in sufferers of ME-CFS. And then from a former PhD student, we've also investigated the DNA virome and illustrating how the viruses in the gut can influence the bacteria, which in turn causes disturbances now, which are becoming quite frequently recorded in sufferers of ME-CFS. And very recently, Krishan in the group has published a review article focusing attention on viruses that hide within the body, and in particular mucosal surfaces, rather than in the blood. And the fact we may be missing viruses that get reactivated because basically we're looking in the wrong place and in the wrong samples. And this slide taken from the review just illustrates this. So where do these viruses that are continuously being associated with ME-CFS, so for example, Epstein-Barr virus, the herpes viruses, enteroviruses, there are known mucosal reservoirs for these viruses where they can hide away from the immune system, for example, in the oral cavity, the nasal cavity, the gut, as well as the reproductive tract. Various viruses are very good at hiding within these tissues, escaping the immune system, but under various conditions, so under stress, for example, getting another infection, these can get reactivated and then they can spill out from these tissues into the blood where they can trigger things like neuroinflammation related to this can then lead to post-exertional malaise and chronic infections. So we're currently looking at the possibility of these viruses hiding in these tissues, particularly in ME-CFS sufferers. And this is not too surprising. This is built on some studies which have shown that viruses can be detected in other samples other than blood samples. So these are viruses that have been identified in saliva or in gut biopsies of ME sufferers. Outside of viruses, there are other infections which are related to ME-CFS, fungal, one which Katharine will talk about in a minute. But there are also bacteria which have been linked as well to symptoms in some individuals. So, for example, Borrelia burgdorferi or Lyme disease. And then there are urinary tract and reproductive tract infections caused by Staphylococcus aureus, which are also linked to this disease. So I will stop there now and hand over to Katharine, who's going to go into more detail about fungal infections and the current research that relates to fungal infections in ME-CFS. Katharine.
Thanks, Simon. Please, can you press next? Yeah. So I'm going to talk about, as Simon just said, the fungal component of potential fungal component of ME-CFS. So let's start with Candida. And Candida is a type of yeast that commonly lives in and on the human body. You can find it in the gut, in the mouth, on the skin, and in the genital tract. For most people, it lives there harmlessly under control by the immune system and by other microbes. However, under certain conditions, Candida can overgrow or cause infection. This happens, for example, after antibiotic use, during immune suppression, with hormonal changes, or when the gut or skin environment is altered. Candida infections can range from local infections to systemic infections that affect the whole body. And even when the infections are not severe or life-threatening, they can cause persistent symptoms such as fatigue, brain fog, gastrointestinal problems, pain and immune disturbances. And these are symptoms that have considerable overlap with ME-CFS. So Candida, it's normally present, but under the wrong conditions, it can become problematic. And I'll now briefly summarize some of the scientific evidence that has raised questions about the role of Candida and fungal infections in ME. Next, please, Simon. So the first study looked at the gut microbiome of people with ME-CFS. And they found that an abundance of Candida albicans in the stool sample was significantly higher in patients who were in an active phase of disease compared to those in remission, suggesting patients had intestinal candidiasis. Intestinal candidiasis refers to an overgrowth of Candida in the gastrointestinal tract. And as I mentioned earlier, Candida normally lives in the gut in small amounts, but problems can arise when it grows excessively or where when the immune system is unable to keep it under control. Symptoms associated with intestinal candida overgrowth include gastrointestinal symptoms such as bloating, abdominal discomfort, diarrhea or constipation, nausea, and also things like fatigue and brain fog. And now again, you may notice that the considerable symptom overlap with symptoms commonly reported in ME-CFS. The diagnosis of intestinal candidiasis is not straightforward, as there is no single gold standard test. When intestinal candidiasis is suspected, treatment offering includes dietary changes alongside other approaches. And the goal of these are not to eliminate the fungi completely, but to reduce conditions that may favour fungal overgrowth. Examples of a candida reduction diet include things like reducing simple sugars and refined carbohydrates, limiting alcohol, and moderating the intake of certain highly processed foods. And also some dietary approaches involve temporarily reducing foods that are naturally high in yeast or mold. One paper in ME-CFS patients showed that there was symptom improvement following one of these tailored diets designed to limit fungal growth, as well as treatment with antifungal agents in ME patients. And the third study was a 17-year follow-up study involving around 400,000 individuals. And these were people with a newly diagnosed infection, including candida infections, and they were matched with healthy controls. And it was found that those who had experienced a candida infection had significantly higher risk of later developing ME compared to controls. This alone doesn't prove causation, but it does suggest candida infections may be a risk factor. Next, please, Simon. There has also been a hypothesis published, and in this paper, the authors highlighted the similarities in symptoms and immune disturbances between ME-CFS and what's sometimes referred to as chronic candidiasis syndrome, which is a condition that doesn't have formal diagnosis, but it's used to describe a situation where individuals experience persistent multi-system symptoms thought to be related to ongoing candida overgrowth or immune response to candida. So again, the symptoms described are similar to ME-CFS and include fatigue, cognitive dysfunctions, muscle and joint pain, gastrointestinal symptoms, and recurrent infections. The paper also highlighted similarities not only within the symptoms, but also in immune disturbances, with patients having natural killer cell dysfunction and T cell dysfunction. And the authors suggested that the fungal-driven immune disruption could make it harder for the immune system to control latent or persistent viral infections, which is particularly relevant in ME-CFS because of the persistent viral infections frequently reported. And now it's important to highlight that this is a hypothesis and it's not a proven explanation, but taken together, these papers give a strong reason to investigate things further in ME patients. Next, please, Simon. So fungi don't affect health just through infection. They also produce something called mycotoxins, which is a toxic substance released as part of their metabolism. And mycotoxins can enter the body through inhalation, ingestion, or skin contact, particularly in environments with mold exposure, and they can affect the immune system, nervous system, energy metabolism, as well as specific organs such as the liver and kidney. Next, please, Simon. Several studies have found mycotoxins in the urine of people with ME-CFS, suggesting ongoing exposure or impaired clearance. One particular report described a family exposed to mold who developed ME-CFS-like symptoms, and when they did follow-up analysis 3 years later, when the family no longer lived in this mold-exposed environment, they still actually had the symptoms persisting. So it raises a question about whether fungal toxins may contribute to symptom persistence in at least a subset of patients. And it's clear to say that further research is still needed, as it's a complex area and not everyone with ME-CFS will have fungal exposure or fungal involvement. But the evidence is strong enough that it makes a case for systematic investigation of fungal infections in ME-CFS. Next, please, Simon. Next, please, Simon. So at the Quadram Institute, we're undertaking 3 separate studies which are investigating how the fungal microbiome, fungal pathogens, and infections may interact with the immune system in ME-CFS, and I'll just go through and briefly introduce each of these projects. Next, please, Simon. Next, please. So the first study is called Compass Me. This is a study that's been set up by Krishani, and we're investigating microbes at mucosal surfaces. So we're specifically using swab samples from the mouth, nose, and gut, and also collecting saliva samples. As Simon explained earlier, these surfaces are important, and this is because they are the key interface between the body and the external environment. So my role within this project is to identify exactly which fungi and yeast are present at each of these mucosal sites and determine how their presence differs between individuals. Next, please, Simon. Next, please. So the second project involves the analysis of blood samples for evidence of fungal exposure and immune responses. So we're using samples from 2 Norwegian studies. Both of these studies are fecal microbe transplant trials, and one of them was undertaken in IBS patients and the other in ME-CFS patients. And we're using the blood collected from this trial in order to analyse the immune response to fungal infections and also other things too. And we're using IBS participants to act as a disease control, which allows us to ask whether findings are specific to ME-CFS or more broadly associated with gastrointestinal chronic illnesses. Next, please, Simon. So as you can see, we're going to use these blood samples to do a range of assessments. Regarding the fungi-relevant experiments, we'll be looking at antifungal antibodies, which are antibodies produced by the immune system in response to exposure to fungi such as Candida. Measuring these antibodies can tell us a number of things, which include whether the immune system has been exposed to fungal antigens, whether there may be ongoing or repeated exposure to fungal antigens, and how strongly the immune system is responding. Now, elevated levels don't necessarily mean an active infection, but they can suggest increased immune recognition of fungi, which may be relevant in chronic illness. Fungal translocation markers is something else that we're interested in looking at, and these are indicators that fungal components such as fragments from the cell wall may be crossing the gut into the bloodstream. When fungi components leak into the circulation, this can trigger immune activation, drive inflammation, and contribute to systemic symptoms. So by measuring these markers, we can assess whether there is evidence of increased fungal exposure to the immune system, even without a diagnosed invasive infection. Next, please, Simon. So this brings me on to the final project, and it's the main focus of today, and this is the Norwegian fungal infection and ME patient survey. Next, please, Simon. So this survey is designed to address a very simple but important question. Do people with ME-CFS have an increased susceptibility to fungal infections? The survey has three main aims. First, we want to determine the prevalence and duration of yeast and fungal infections amongst people with ME compared to healthy controls. Secondly, we aim to identify lifestyle and environmental factors associated with increased susceptibility to fungal infections. And these include things like medications, living conditions, and other exposures. And finally, we want to explore whether these risk factors differ between ME-CFS patients and healthy controls. And we will use this information to help guide our future research and also identify areas that need deeper investigation. Next, please, Simon. So for this survey, we are aiming for 1,000 responses from people with ME-CFS and also 1,000 responses from a control group without ME-CFS. The survey includes questions about yeast and fungal infections at different body sites, medications that can increase the risk of fungal infection, lifestyle habits, and environmental factors that are known to influence fungal risk. And I want to just emphasize at this point that we're very interested in anyone with ME-CFS to take part, including people who've never had a fungal infection, because this information is just as valuable and it allows us to have a clear overview of the exact prevalence of fungal infections in ME-CFS. Next, please, Simon. So the questionnaire was developed in multiple stages. We first drafted questions based on the Centers for Disease Control and Prevention guidelines on fungal infections. We also met with an epidemiologist in order to refine our research question and then target the questions within the survey to make sure they answer the question of interest. We had patient and participant involvement in reviewing the questionnaire. We consulted with a clinician, and with all these, we then did modifications, taking everything into account, and then Trude translated this into Norwegian for you all to answer. Next, please. So if you're interested in taking part, this QR code, if you scan it, you're able to access the survey, and just a few instructions for how to complete the survey. So this survey is anonymous and it is limited to one response per device. So if you have more than one person in a household completing the survey, you'll all have to make sure you use different devices, and you can complete it in several sessions so long as you don't delete the cookies in between sessions until you've completed the survey. Next, please, Simon. So thank you all for your attention. We're happy to take any questions, and we would just like to acknowledge both the current and past members of the Quadram ME-CFS team, and also all of the research donations from charities and grants, which have been very generous and received gratefully. And then also, as Simon mentioned earlier, we have many collaborators throughout the Europe. Thank you.
I'll just leave this up just in case anybody still needs to access the QR code.
Thank you. Well, I must say it's been very interesting to been allowed to be a part of this project. Katharine and I have had a long discussion about questions and how to ask them, and I think we have an interesting survey that I hope many will fill in. We're also going to put some information on the ME Association website, so you can be able to find it from there. There are no questions yet, but I have one that's a little bit on the side of the main presentation. But Simon, I would like to ask you a question, if I may. You have listed a lot of different triggers and infections for ME/CFS. Recently, a lot of people get ill after a COVID infection, and there's some studies that find a lot of similarities between ME and COVID, long COVID. But there are also differences. Do you think there are differences between people who had different pathogens as triggers?
I think the answer to that is probably yes. Because I think something that we often keep ignoring is that the disease is very heterogeneous and there are different clinical subtypes, each of which has its own pathway of disease progression. So it's entirely likely that there are different pathways that are activated in response to different pathogens. And we're particularly interested in one particular type of virus that lives within our body. It's part of our genome, so the genetic material of the virus is integrated into our own genome, and these are called endogenous retroviruses. So up to 8% of our DNA is made up of these viruses. And normally they are not important, they don't cause any harm, but they can get reactivated and then they become dangerous. And they have been increasingly linked to diseases such as multiple sclerosis, type 2 diabetes, and maybe some dementias. And one of the things that triggers their reactivation are other virus infections. So, for example, SARS-CoV-2, influenza virus, Epstein-Barr virus, all these viruses can reactivate this endogenous virus, and then that causes disease. So depending on which family of viruses are the trigger and which family of endogenous retroviruses respond to it, we can get different clinical phenotypes, if you like, that maybe not entirely overlap. And because these endogenous retroviruses live in lots of tissues, again, the tissue in which they get reactivated may cause unique symptoms that are specific to that tissue, be it in the muscle, be it in the brain, be it in the immune system. So depending on where they are activated, you get quite different features. Sorry, that was a bit long as an answer.
No, that's very interesting. I'd just like to have one follow-up question, that if there are different pathways from different pathogens, then conceivably you could have more than one. Would that be correct? I mean, I'm talking from my own experience. I don't know what triggered my ME, but I know that 10 years later I got parvovirus, and I then got symptoms that haven't disappeared afterwards, and I became a lot worse. So could you say that if you had ME from one trigger and then, for instance, you got a fungal infection, that you have one thing on top of another?
Very much so. So I think because all these infections lead to one endpoint generally, which is your immune system. Your immune system has to react to it. And if the immune system is overstimulated in trying to respond to more than one type of infectious agent, it can just lead to this chronic ongoing immune activation, which then spills over into inflammation in all your tissues. So the key is removing the trigger, and that may not be one, it could be 2, 3 even. So if we can eliminate the virus, the fungi, the bacteria, that should stop this downstream cascade of immune activation, which leads to all sorts of difficulties and problems. So yes, these things all amplify each other, or can amplify each other.
Thank you. That leads very nicely into the one question we have received here, and it's She says, I became ill after mononucleosis, but it took 15 years before I was diagnosed with ME. In the meantime, I saw a private doctor who treated me for candida, including through diet, medication, and avoiding damp environments when possible. I actually felt quite well for a time, but then I got worse again. Later, I have thought that the diagnosis may not have been correct, but that some of what I did helped. For example, I also have IBS, and diet can help with that. I also have outbreaks of fungal infection on my nails and in the genital area, but I treat these with over-the-counter products. Based on what you are saying here, could it be worth trying treatment for candida again?
So I should predicate what I'm about to say by saying I'm not a clinician, so I can't really give medical advice. But yes, because fungi, I mean, Katharine can back me up on this. Fungi are very, very difficult to treat because a lot of the current medications used, the fungi can develop resistance to, and they can cause these chronic low-grade infections of nails, for example, skin. And if eventually they just overcome all possible drugs and treatment, that's when they can potentially be very serious. So yes, there is a smaller number of drugs available to treat fungal infections than for bacterial or viral infections. I mean, Katharine, do you want to comment any further?
Just echoing what Simon said, in that I'm not a clinician, so I can't give medical advice. And without having any tests done, it's hard to kind of hypothesize what's occurred in your situation. It could be worthwhile maybe going back to your GP and discussing trialing the dietary changes that you made, because if you experience improvements in them for whatever reason, whether that be the candida overgrowth or whether that be the IBS, I think it's worth exploring.
Thank you. There was one thing I thought maybe we should say a little bit more about. It's the why you're doing this survey in Norway. And as far as I understand, that's because you already have the fecal samples and blood samples from Norway, from the comeback study, and that you want to compare those to the survey answers. Is that correct?
Yes, that's correct. And environmental factors play a key role.
That's all right. Maybe I'll just say that, having worked with Gertrude and with Christian on FunCap, we were very impressed with how organized patient support group in Norway is. And so it was a natural place to go when we wanted to conduct a survey among ME sufferers.
Thank you. That's, yes, we have had quite a lot of success with our surveys, and we're very grateful to all the patients who are so eager to answer and help us document their experiences. So that's, I'm hoping that we'll get a good response to this one as well. Then we haven't received any more questions, unless there's anything you want to add at the end, Simon or Katharine?
I just want to again emphasize and encourage everyone who has ME-CFS to complete the questionnaire, regardless of whether or not you think you've had any fungal infections, and because that helps us give a true picture of what's going on. And if you know anyone without ME-CFS too, who can complete the questionnaire, they can use the same link. There is options within the questionnaire which direct you to questions for if you're an ME patient or whether you're a healthy control.
Just for practical purposes, as I said, the survey is limited to one answer per unit, but if you delete cookies in the meantime, you can answer one more time. Because that's sometimes a problem for families if they use the same computer. but it's possible if one person finishes, delete cookies, and then another person can answer the survey. So ask family members, healthy family members, to answer as well. Simon, do you have anything you want to add at the very end?
No, I'd just like to thank you all for giving us the opportunity to present our work to you today, and thank everybody and anybody that wants to contribute. It's very valuable. It informs our research, it guides our research, and it can prove to be very insightful in terms of how these pathogens cause disease and then direct individuals affected to more evidence-based treatments that have a better chance of success.
Thank you. And then I hope that in a couple of years' time, we can invite you back to give us the results from this survey and the study.
Sure.
Okay.
Glad to.
Well then, again, thank you, and thank to everyone who joined us today. And please answer the survey. And this will be recorded, and it will be posted to Facebook and YouTube as soon as we are able to. So thank you and goodbye.
Thank you.